When we talked about getting an unknown bacteria, I don't remember if I told you there are 9 possibilities as to what that bacteria can be. Right now, in face-to-face class, we are doing a series of experiments that are giving us more information about how each of these 9 bacteria react with specific tests. Exercise 11 deals with selective and differential media. If you remember back, selective and differential media from lecture is a type of media that reacts a specific way. Some only grow one type of bacteria and not the other, so gram-positive and not gram-negative, or vice versa, gram-negative and not gram-positive. Some of them actually react with specific things on the bacteria, causing color changes that also show up in that media. Let's talk about what we're going to do in this experiment. Again, what's the purpose? It's used to differentiate between various strains of bacteria. Every bacteria will not react the same way based on years of research. Selective means that it will grow one strain but not the other. It'll grow, again, gram-positive but not gram-negative. In this experiment, we will use 3 different types of selective and or differential media: McAllisters Auger, EMB Auger, and Pea Auger, which I'll tell you what the names stand or the abbreviations stand for in just a second. We have 3 different types of augers: McAllisters, EMB, and PEA. 3 augers have specific characteristics that allow them to be selective. Only gram-negative or only gram-positive will grow on top of that auger. Differential means that when you have the gram-negative bacteria growing on there, maybe something changes color in a specific way to indicate a specific trait for that bacteria. Let's talk about McAllisters Auger first. McAllisters Auger is both selective and differential. It is selective for gram-negative bacteria. It doesn't want gram-positive to grow on top of it. It also is differential in the fact that you have a pink color pop up if there is lactose fermentation. So it's a characteristic of the bacteria that changes something on the auger that's a red flag for us to go, oh, this bacteria does this. Don't forget, every time we have a trait that is unique, it's something to add to the list of characteristics that give us an idea of what we're looking at as far as our 9 possible bacteria for our unknown. McAllisters. It is selective. It inhibits gram-positive, so it's selective for gram-negative. And it is differential. It'll turn pink with lactose fermentation. Remember, fermentation means it'll use lactose as a fuel source. It'll break it down and make ATP out of it. Next, we have EMB, eosin methylene blue auger. Why is it called that? Because it has 2 pH indicators, eosin Y and methylene blue, and they can combine to form a dark purple color when in the presence of a low pH. That's differential. Only certain bacteria have a low pH and they'll show up dark purple. It is selective, however, in the fact that it inhibits gram-positive bacteria. What does that mean? it's selective for gram-negative. It likes gram-negative. So EMB is selective for gram-negative as well. It likes gram-negative. Differential. Dark purple colonies means lactose fermentation. If there is no lactose fermentation, we get clear colonies. They look like glass, so no fermentation. Another differential trait that we have with EMB is exclusive to bacteria that have flagellum, that move around with their flagellum, and have very strong acid byproducts. These two things together actually interact with the ESNY and the methylene blue, and it turns the colony into a very distinctive color. It looks like green aluminum foil. It is metallic and it is lime green. Here. Now for us, the only type of bacteria that we have that does this is E. coli. The problem with that is E. coli isn't the only one that has flagellum and strong acid end products. With that in mind, our pool of 9, E. coli is the only one that does this. So when you put E. coli on EMB auger, you get this metallic green colony formation. But it's a differential for flagellum plus strong acid byproducts. EMB is selective for gram-negative, and it's differential. If you've got purple colonies, lactose fermentation. If you have metallic green, well, for us specifically, you have E. coli, but in reality, this is what it's differential for. It's the flagellum with the strong acid byproducts. The last auger that we're going to discuss and actually use in the lab today is PEA, phenyl ethyl alcohol. PEA is just easier to say than phenyl ethyl alcohol. This is the only auger out of the 3 that selects for gram positive. The other 2 are selective for gram negative, both McConkey's and EMB. PEA is selective for gram positive, meaning if I'm a gram negative bacteria and you put me on PEA, I will not grow. Why? I can't make my own DNA, which means I can't make copies of myself. I can't divide. I can't do anything as far as making more bacteria because the PEA prevents me from doing it. The only exception are going to be negative obligate anaerobic bacteria. We don't have chambers with no oxygen here at CBC. So nothing that we have out of that pool of 9 belongs to this category. The only thing that we have that will grow on PEA has to be gram positive.